This is episode 4 of the Immunology podcast, Therapeutic T-Cell Engineering, with Drs. Carl June and Philipp Rommel. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Roud. Welcome back to the Immunology podcast, where we have amazing conversations with immunologists. The Immunology podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. Today, we have Drs. Carl June and Philipp Rommel from the Perelman School of Medicine at the University of Pennsylvania on the podcast to talk about their research to developing new ways to enhance the ability of the natural immune system to recognize and eliminate tumor cells. We've also got our usual roundup of recent highlights in immunology news coming up, but first...
Ensure reliable results with your immunology research. From primary human cells to cell isolation kits, culture media, supplements, and antibodies, STEMCELL Technologies provides the tools you need for every step of your immunology research. Interested in cell isolation? Use EasySep to isolate highly purified immune cells from virtually any sample source in as little as 8 minutes. Cells are viable, functional, and immediately ready for your downstream applications. Learn more at easysep.com.
Well, hey there, Brenda. How are you?
Doing great. How about you?
Oh, pretty good. We're getting ready for the summer, so that means we've got to plan out all the summer camps for the kiddos.
I heard that there's some plans there, some contingency plans for summer camps in the U.S.
Yeah, we actually have them this year. We're not just sitting the kids in front of the TV. We're upscaling our parenting.
It's been raining for days at end here, so I kind of envy your idea of summer. Okay, why don't you tell me about today's papers? I'm very excited to hear what research you've been looking at.
All right, yeah, let's dive right into it. So I'm going to start with a very interesting CRISPR screen. So this paper was put out in Science Advances. It is titled Pooled CRISPR Screening Identifies M superscript A as a positive regulator of macrophage activation. It is by first author Jiu Tong and last author Huabing Li. So this was an interesting paper in that it started getting at a lot of what we look at in terms of mRNA regulation and really diving deep into some processes that hadn't been really appreciated before. So M6, and that's little m superscript 6, regular size, capital A, RNA modification is something important in a lot of cellular processes, and it helps regulate cellular degradation. Or not cellular degradation, helps you regulate RNA degradation. And so how it's important in some processes have been understood, but what it means in terms of this pathway in immune cells, and in particular innate immune cells, is poorly understood. So I'm not an expert in this pathway, so I had to do a little bit of reading here to understand it. And there's writers and readers of these RNAs modifications. And so this paper really, through its work, focused on the fact that it was a M6A writer that was important here. And what they did is they did a CRISPR screen to look at macrophage activation by LPS. So that was their model system, looking at macrophage activation by LPS and understanding how this pathway was important in that stimulation. And so what they found was that this one gene, MET-L3, and that's METT-L3, which is a writer of this modification, was critical for macrophage activation. And so the way it works is that if you knock it out, these mice with a conditional knockout, so a lyse-M-Cre transgenic system, if you knocked it out, mice were susceptible to bacterial infection and had faster tumor growth. And so you kind of deactivated killing functions and protective functions of macrophages because of decreased LPS activation. And what they showed is, and they did a very nice signaling cascade here, is they said, okay, so this gene, if you knock out MET-L3, it is affecting mRNA modification. That's what it does. What targets of it matter, right? Because what you're basically doing is turning off the ability for an RNA degradation pathway to occur. So you have your transcription, and once things are transcribed, they need to be translated, but the steady state of the mRNA is going to dictate a lot of pathways. And so this gene is responsible for targeting by writing on it and making this mRNA modification something important downstream for it to be targeted for degradation. And so they said, well, okay, so this is really important. What genes are involved? And this is what really bumps the impact factor up here, makes it really interesting, is they found that its deficiency in the writing gene, that MET-L3, led to loss of the RNA modification on IRAKM or IRAKM mRNA and slowed down its degradation. So you had higher levels of IRAKM, which is part of the NF-kappa B TLR signaling cascade. It kind of sits downstream of MyD88, so right underneath the adapter, and helps target NF-kappa B regulation and translocation. And so it turns it off. And so then you have decreased TLR signaling. And so in the absence of this, you have higher levels. If you knock this out, you have higher levels of this protein and thus lower levels of TLR activation. And so they did a really good mechanistic deep dive. It's really powerful that way, which I appreciate. I appreciate NF-kappa B signaling coming from an NF-kappa B lab in grad school. And so it's nice to really see how this played out and that they were able to actually get down to an RNA level. And so they did a lot of cool experiments where they made mutations in upstream in the 5' and 3' UTRs that would inhibit this ability for it to write. And so those would rescue things because now you wouldn't have the modification. You would have the modification. So they could basically switch it on and off the effects and recapitulate it by modifying the upstream and the 5' UTR and 3' UTR to make it unable for this enzyme to work. And so if the enzyme couldn't work, then it mimicked what would happen in the knockout. And so they could recapitulate the mechanism that way. And so there we go. I thought it was a really nice mechanistic immunology paper. If people are curious, the way RCAM works is it's induced by the activation of macrophages and prevents the dissociation of IRAK and RAK4 from MyD88. So it inhibits the formation of TNF receptor-associated 6 complexes and so suppresses NF-kappa B activation. So it's right downstream of MyD88. So your TLR activates MyD88. MyD88 then makes complexes, which then leads to eventual NF-kappa B translocation, and so inhibits one of the high upstream complexes.
So in a natural environment, where would you need, or where would this pathway make sense? Because then what it does is dampens the activation of the macrophages.
So it basically keeps them somewhat quiescent or turned off at baseline. And so it's kind of, this writer is there present at all times so I guess what happens is that in activation, this writer activates to target the inhibitory pathway for degradation.
All right. That would be maybe to just contain the activation response, that you prevent an overactivation, for example.
Well, kind of. So in the knockout, you don't have activation anymore. So in the knockout, you don't activate, right? So the knockout mice have worse response to infection and worse response to tumors. So their tumors are bigger. And so you have lacanate killing. And that's because you can't degrade the suppressive pathway.
Right.
Yeah. And so the suppressive pathway stays activated even when you're trying to turn the system on, essentially. And so this is looking at the regulation of that. This is understanding that LPS, in addition to all the other things it does, activates this enzyme, the metal-3, to then degrade various proteins that inhibit NF-kappa B activation.
I'm always amazed at the multiple levels of regulation that these responses have. And they're also, it's kind of redundant. There's so many different layers. So I was not aware of this, for example, this type of RNA modifications as a mechanism for regulating gene expression like this. It's wow.
Yeah, just the fact that there's more and more. I mean, I think that's the whole point. What we've realized with the Human Genome Project, right, is we don't have nearly enough genes that they think we should. But there's all these, either miRNAs or different microRNAs, different 5' and 3' structures that are important for regulating genes, different pathways that regulate their activation. So you can get a lot more done with less genes by regulating everything with a bunch of layers on top of each other.
Yeah.
So make the system more complex per gene versus increasing the number of genetic complexity. It's really interesting.
Very nice. Well, I mean, in the topic of, regulation and layers of regulation, I have also a story about regulating T-cell activation by unconventional pathways. And so I want to talk about a study that looks at the recognition by T-cells of particular peptides that are derived from cytomegalovirus, or CNV, that are presented on a non-classical HLA-1 molecule, which is HLA-E. And this paper comes from the lab of Andrew Brooks at the University of Melbourne, Australia, of course, and first author Lucy Sullivan. It was published in Science Immunology recently, and is titled Natural Killer Cell Receptors Regulate Responses of HLA-E Restricted T Cells. So, for our listeners who don't remember, so HLA-E is what is known as a non-classical HLA-1 protein, and the main thing this immunoglobulin presents is peptides that are derived from the leader sequences of other HLA molecules, HLA-1 and HLA-A and HLA-C. And this HLA complex, together with the leader peptides, are ligands that can be recognized by receptors that are traditionally expressed on NK cells and can either inhibit or activate the NK cells. And the idea is that these HLA-E molecules are a mirror that report on the expression of HLA-1 molecules, and it's a way of the external environment to look and, for example, recognize that if, for example, a pathogen is downregulating the expression of HLA-1 molecules to kind of stay away from immune surveillance, this will also affect the expression of HLA-E on the surface and then signal to, for example, NK cells. And this would allow, for example, if enough signals are available, NK cells to activate and lyse and attack this potentially infected cell. And so there's this one protein in CNV. So CNV, I mean, as you know, is a very prevalent infection on human populations. About 50% of people have latent or are seropositive for CMV or have latent infections. And among those people that are seropositive, you can see that they have a lot of T cells, a lot of memory T cells that are specific for CMV antigens. So it's really amazing how CMV can really affect the composition of your T cell compartment very, very strongly. And there is one peptide derived from a protein UL40 from CMV that actually can bind to HLA-E and kind of mimics this HLA-1 derived peptides, this leader sequences. And the idea is that it can kind of prevent the downregulation of the HLA molecule on the surface and replace the actual endogenous peptides. And this can be can be used as a strategy by CMV to, evade immune responses. And what is very interesting is that this peptide is very, very similar to the endogenous peptides. But what you see is that people have, particularly seropositive people, have substantial T cells that are recognizing this UL40 peptide bound to the HLA-E molecule, which is kind of strange. And when you think that most of the times this molecule is presenting self-antigen. So the question is, how does the body make sure that the recognition of this viral peptides does not cross react to the endogenous peptide? And so they look that because these peptides are very, very similar. So what they do is they look into the receptors of these specific T cells, and they really study the specificity of the receptors and the characteristics of the T cell receptors for these cells, and they find that many of them actually are potentially cross react to the endogenous peptide being presented on this HLA-E molecule. And then that begs the question, how does the body keep the autoreactivity from happening? Especially in these people where you have a lot of these T cells that can potentially recognize this antigen in your blood at all times. And what they look is that there is the expression of these receptors that are usually found on the surface of NK cells, and they are usually part of NK signaling and activation. but they are also expressed by these T cells. And they basically look at two groups of TCR receptors, one that are fairly low affinity for endogenous. They have a higher affinity for the viral peptide than from the endogenous peptide. And those have on top of that, they have activating receptors that can also bind to the HLA-E molecules. And that together with the low affinity TCR allows the T cell to actually activate and attack an infected cell. And then there's another group of cells that have high affinity receptors, which in principle would be a higher risk of autoimmunity, of attacking our own antigens. But then these cells are expressing, on the other side, inhibitory receptors that are usually found on NK cells. And these receptors prevent autoimmunity in a way that allows for the balance and doesn't result on self recognition of the peptides that are loaded with endogenous. So the HLA-E molecules loaded with endogenous peptides, although they're very, very similar. So I would say that the really interesting thing here is that they see that they use this KIR receptors that are mostly found in NK cells, and they show that they are actually modulating this response from the T cells that are recognizing peptides that are very, very similar to endogenously produced peptides, and they modulate and they prevent autoimmunity or help the recognition of the right peptide under conditions that are compatible with that viral infection. So I thought it was very nice, very kind of a little bit outside of the box when you think of presentation by non-HLA A, B, or C. And it was really, really nice. I really recommend anybody looking at this topic to check this paper. It was very, very nice paper to read.
So you're saying that there's T cells with HLE on it that are restricted, that have self-antigen, and these T cells that are presenting by HLA-E self-antigen have an NK receptor?
No, it is T cells that have TCRs that recognize this particular ACMV peptide in the context of HLA-E.
On a regular host infected cell.
Exactly.
But they also have NK cell receptors.
Right. And those receptors make all the difference to TCRs that have low affinity, but they have high specificity for the viral antigen. And they help those cells activate further when the cell is actually infected. And then you have TCRs that have less specificity but are higher affinity. And these receptors, they have different NK receptors that actually are inhibitory, and they prevent autoimmunity. So they kind of swing both ways in the different types of cells. They don't really say how these cells acquire these receptors, but they're there and they're modulated in their function.
And that's really interesting. That I think it shows the plasticity of immune cells.
Yeah.
And that they have a whole bunch of different receptors that just show up that are on other cell types when they need to have them.
I know, right? Yeah. And also the peptides are very, very similar, but they're so important that it makes sense that you have to find a way of differentiating them somehow, or like preventing, or manipulating the activation accordingly. And I thought this seems like a very smart idea. Cells are very smart.
Yeah, that's fascinating. All right. Well, I guess we'll jump on to my next one here. We're going to play a little quiz, but I'm going to give you the title of it first. So this paper is Notch4 signaling limits regulatory T cell mediated tissue repair and promotes severe lung inflammation and viral infections. It's in Immunity. First author is Hani Harb, and last author is Talal Chatila. And so again, I'll read it one more time. Notch4 signaling limits regulatory T cell mediated tissue repair and promotes severe lung inflammation and viral infections. So I picked this to start looking at because, wound healing, tissue repair, T cells, all things I'm very interested in. COVID. It's back because it never went away. This is actually a paper about COVID, which I thought was really interesting. And so I'll give actually what I think happened here, and we know how science works. They were studying this in influenza already, because that's what most of their modeling is in. And then the authors probably bookended it on either end with some COVID studies. So they were already studying these pathways that are very interesting, and then probably did a COVID study to show that this is also true of COVID, not just influenza. I mean, kudos to them. They figured something awesome out. They related it to the pandemic, but I think this is actually probably a more universal setting and they show that than just COVID. It's just also relevant to COVID. So the high level things is that, and what they showed here is that Notch4 signaling or Notch4 expression on Tregs is associated with increased COVID severity. Notch4 inhibition suppresses lung inflammation in viral models, aka influenza, and then just IPIC stimulation in mice. And then they got to the mechanism of this is that what happens is Notch4 inhibits a molecule called amphoregulin, and that amphoregulin is produced by is in the Treg mediated tissue repair function cycle. And so what happens is that Tregs will release amphoregulin, which leads to tissue repair and a shift upwards of M2 versus M1 macrophages, and that's kind of known signaling. But what they're showing here is that in viral infections, you have an IL-6 cascade that activates, an IL-18 cascade that activates, and that you then, or sorry, not IL-6, IL-18. IL-18 is turned off. So you shift normally, you have high levels in a viral infection, you have high levels of IL-6 and less IL-18. That leads to Notch going up. Notch inhibits the IL-18 cascade. Amphoregulin goes down, and you end up with an M1 type. And so what I thought was really interesting about this was they linked something to a very important biomarker of Notch4. It's important in innate cell regulation. So you can kind of, if you want to put your stem cell hat on for a second, you can think of beta-catenin and those pathways as one. You can think of YAP-TAZ, so the hedgehog, and signaling is another set. And then you can think of Notch as another kind of big bin of regulatory processes. And so what they were able to establish was that it's really the Notch driving this in this setting of viral infection and really kind of steering. I screwed up. YAP, it's not hedgehog, it's hippo. It's the other H. And so you have like YAP, hippo pathway. You have Wnt beta-catenin pathway. And then you have Notch. And so there's three major accesses of downstream kind of shifts in state, whether you're thinking about stem cell differentiation or immune activation or shifting in pathways. And they were really able to show that in this case, it's the Notch4 that's promoting the innate immune cell activation by downshifting. So its presence then shifts you to that hyperinflamed M1 state and decreases tissue repair. And then that leads to worse outcomes. And so they show that the viruses essentially do this. So COVID does this. COVID upregulates IL-6, which we know about. It then, they don't go through all these mechanisms, but it then inhibits IL-18. It's Notch dependent, so high levels of Notch are necessary for the inhibition of IL-18, and then amphoregulin drops. And so because of less amphoregulin, you switch from M2 to an M1. And then normally you have more M2 in a tissue repair state. And then they showed that IPIC, an in vitro polypeptide mechanism to stimulate cells does this. So poly-IC. So for those who don't know that, polyinosinic polycytosidic acid. So for those who may not be familiar with this is a very common toll-like receptor 3 stimulation. It kind of mimics general viral RNA sensors and induces all those downstream pathways. It's a very common in vitro tool used in research to stimulate cells. And then they show that flu does this in mice too, essentially. And they tie it all up.
But they show, so the Notch4 expression is in Tregs. So it is this effect is through Tregs.
Right. So it's all happening in Treg, and Treg secretes this thing called alpha-regulin, which then goes and affects the M2, M1 state of a macrophage.
Okay. So yet another way Tregs regulate or kind of translate the signals, because this has nothing to do with T cell activation or TCR signaling.
Correct. Yeah, it's all okay. The Treg. It's all the Treg playing quarterback again, which is what Tregs do.
Another hat.
Yeah, I mean, well, it's the same hat, right? It's not a new hat.
No, okay, yeah, but it's yet another way that T-Regs are getting information from the environment.
Right. Yeah, and I think that's a key thing that keeps coming back, is that these Tregs kind of sit there and can integrate a lot of signals. And of course, then there's the innate equivalent of a Treg, which is an MDSC, myeloid-derived suppressor cell, which we still don't know as much about as we want to. But it's interesting that it's this notch that's doing it here.
We can even dial up a notch more, because I have another notch related story for you.
I appreciate the dad joke. Thank you.
Well, somebody has to do it.
Yeah.
But this is a little bit different, because this is a synthetic notch story. So this is a story about my favorite immune cell type, which are synthetically, genetically modified T cells. And this story comes from the lab of Cole Royal at UCSF. And the first author, Axel Hieronymus Wittsten, and also recently published in Science Translational Medicine, back to another similar paper that I will just quickly mention later. And it's titled SynNotch, also synthetic notch CAR circuits, enhance solid tumor recognition and promote persistent anti-tumor activity in mouse models. And this is a very cool story about synthetic biology at the service of kind of therapy, hopefully in the future. You mentioned notch, and I think something that maybe I just want to clarify is that notch receptors are very cool, because they work by, they have a receptor outside in the extracellular part, and then inside the internal part of the receptor gets detached from the surface and can translocate to the nucleus upon activation of the receptor outside. So basically, you can, by activating the external receptor, you can result in the translocation of, for example, transcription factor, and then you can start some transcription program inside the cell. And so what this model does, the synthetic notch receptors, basically exploit this function of the notch, and they replace the external or part of the receptor by, for example, a single chain fragment from an antibody that can bind to a particular antigen, and they put inside some transcriptional activator of their choice, and thus they can make a little signaling module. And so based on this, this work shows like a principle, like they show the proof of principle for a system in which they combine in a circuit a synthetic notch receptor that can bind and recognize a particular antigen that they have found that is very highly expressed in their tumor models that they study, which is mesothelioma and ovarian cancer. And this antigen is called alkaline phosphatase placental-like 2, ALPPL2. And they combine this with a CAR, chimeric antigen receptor, which we're going to hear a lot about today, that can recognize some other tumor-associated antigen. And they are, for example, in this paper, they look at MCAM, mesothelin, or HER2, which are all known antigens. And they show that they can induce the expression of the CAR with the synthetic notch receptor, and T cells that have this circuit will only express the CAR when they find a tumor that is expressing, in this case, ALPPL2, and they will only respond and will only kind of attack tumor cells that are expressing both antigens at the same time. So it's a really cool concept, because that really is addressing two main issues that we have with CAR T cells in solid tumors. On the one hand, the difficulty of finding a single receptor or a single antigen that is good enough, that is specific enough and expressed enough to target in solid tumors, which is very hard to find. And often they for example, a very sad example is a test of a CAR against HER2, resulting in the death of the patient that got it, because HER2 was expressed also in the lung. And so it's very hard to find this target. So now you can use 2 targets instead of 1, and that will help you prevent attacking normal tissue. And on the other hand, another part is the constant expression in those CAR T cells that are driven by promoters that are constantly activated. These CARs have this tonic activation, this tonic signaling that result in the kind of exhausted phenotype and a reduction in the survival of the cells, particularly within a tumor microenvironment, and makes these cells in the end not persist enough and exhaust before doing the job. And thus, by using this inducible system, they prevent these two problems. And kind of long story short, they really show both in vitro and in vivo that this system really has a lot of potential for solid tumors and CAR T cells for solid tumors. And also in a back-to-back paper from a related lab, they also look at the use of this system for treating glioblastoma, which is a very, very hard brain tumor to treat. So I think this was a fantastic read for anybody looking for synthetic biology and immunology. I really enjoyed it. So I also was kudos to the authors and to the labs involved. Great job.
So I guess as the resident CAR T cell therapy nerd here, Brenda, does this package that they need to do with A, mutate Notch, right? Because that's part of it. And then B, also, or introduce the Notch receptor and also do a chimeric antigen receptor. Can you fit that in a single lentiviral package? Is this at a what level where it can be commercially done, or are they like doing a double lentivirus and getting 1% yields? And so that's a hurdle. And we need like someone like Philipp Rommel, who we'll be talking to in a little bit, come in with his better technology to get bigger stuff into cells.
So I think they do fit these 2 receptors in one package. I have not, to be honest, I did not look too closely into the manufacturing of these cells.
But they shoved it in one, which I guess that's a good first spot. Okay, that's good to know. Yeah, I'm wondering, like, because obviously when when we're in the science land in academia, we can come up with really cool things, but getting them into people is a whole other set of challenges.
Of course.
Well, we're going to dive in here soon into an awesome set of interviews. But first, time to talk to your sponsors. Decorate your lab with the Nature Protocols wall chart outlining the production of CAR T cells for therapy, from apheresis collection and T cell enrichment to gene modification, expansion, and delivery. Request a free copy of the wall chart at STEMCELL Technologies T Cell Therapy Resource Center by visiting www.stemcell.com/T-cell-therapy.
Today, we have the pleasure to talk to Dr. Carl June and Dr. Philipp Rommel. For many of us, Carl is a scientist that needs no introduction. He is a professor of the Department of Pathology and Laboratory Medicine, the director of the Center for Immunotherapies, and the Richard W. Vague Professor in Immunotherapy at the University of Pennsylvania. And he's a pioneer in the development and clinical application of cell therapies. And we're also talking to Philipp Rommel, who is a postdoctoral researcher at the University of Pennsylvania School of Medicine at Carl's lab. Thank you so much for joining us today.
I guess we can dive right in because you guys are giving us kind of a cool sneak peek on some stuff that you've been working on, Philip. So I remember from some of our time together back at Penn on your T32s, you've been working on kind of next generation CAR T cell therapies and some more precision tools and kind of some neat stuff there. So I was wondering if you could kind of give us an overview of what your mad scientist activities have been with CAR T cell therapies.
Mad scientist, yes. I always say at the end of my lab meetings that I thank Carl for letting me work on my crazy ideas. So that sums it up pretty well. So the June lab itself, we're all working on basically making CAR T cell therapies better for solid tumors and for a variety of other diseases. My project, or one of my projects, was to enhance the CAR T cells using synthetic biology, and there are certain ways how you could make the CAR T cells more potent. My idea was that, the current treatments, they work great for specific type of leukemia and lymphoma. But with solid tumors, a lot of things are basically different. You have to overcome, the hostile tumor microenvironment. You have a lot of basically factors that are different once it's a solid tumor. And my hypothesis was that the next generation of CAR T cells has to be, engineered in a way that it can handle these complex new environmental factors. So my idea was to, initially I basically thought, okay, let's make the CAR T cells very, very potent, and let's just have them secrete, a very potent payload that is very toxic, so that it can kill the tumor cells. And the toxin should be something that is not, natural to the T cells, so that the tumor cell, has never seen it, is not resistant to it. The problem with that is that obviously if you just have the CAR T cell produce a toxin, it will kill itself. So you have to first come up with a system that can regulate the payload release in a targeted way, especially only in the tumor microenvironment. Otherwise, you will have systemic payload release and you will get toxicities. So my first step was to kind of design secretion systems that allow me to release payloads in localized in the tumor microenvironment. And I've designed several different payload systems that work. Some are less good, some are more good, some have more leakage, some have less leakage. So some technologies that are out there. And then the second step was to kind of then come up with payloads that I could use and release inside the tumor microenvironment to kind of kill the tumor. And it sounds a bit of maybe simple, but it's kind of tricky, because everything that once you make the CAR T cells into biological factories that secrete actually at these payloads, you stumble upon a lot of issues. Some of the things that you want to, maybe that you could use to kind of, kill a tumor cell, they are toxic to the T cell itself. So you have to kind of shield it so that the T cells is protected. You might run into issues of production, secretion. So there are a lot of things that you have to kind of engineer to actually make it work. But now I'm at the point where I have secretion systems established, different versions, and now I'm just doing screens for the best payloads to hopefully select my lead candidates and then go in vivo to do some mouse studies there.
I guess that these are the different ways that we're looking into improving our current CAR T cell therapies. Carl, you have been one of the leading scientists in starting CAR T cell therapy, and you actually have your first patient was treated over 10 years ago, if that's correct. How do you think the research that Philipp is doing or other people are doing in your lab, or I think around the world, goes into addressing the main issues that we see? Where are the frontiers in CAR T cell development that we want to look at?
Thanks, Brenda. So, clearly, we now have proof of concept that an engineered cell expressing a foreign molecule can survive for a decade in a patient. And that cell is, genetically modified, and it behaves in a way that was intended. That's a huge issue, because in the past there have been the issues of efficiency. Could you get enough into a patient that it would make a clinical difference? And people were really afraid of a Frankenstein-like cell that would mutate and go out of control. And another issue is that, our immune system is so sophisticated that it would reject any cell that was genetically modified. So we now have, on the basis of this decade of experience, answers to those questions, which is, it is possible to remove the cell from the body, engineer it with one or more genes now, and then re-implant those cells, and they, this aspect of synthetic biology, they carry out actions that the natural immune system cannot. So we have to remember that with checkpoint therapies, which have been a huge advance, that maybe only at best 20 or 30% of patients usually respond. So there's a glass ceiling there that we need to get through, particularly in solid cancers. And what Philipp is doing is extending this out with smarter cells that have different modules that can, go really against the vulnerabilities of a complex solid tumor microenvironment, which is really quite different than the hematologic malignancies, which are really often due to a few gatekeeper mutations that allow uncontrolled proliferation. The solid cancers are much more complex in their cause and evolution.
Is there a specific solid tumor you guys think is a good first test case, or one that really needs this therapy, or that, the Philip, your payloads particularly seem a good proof of concept to go after? Obviously, if you can't say because you don't want to spoil the story, that's okay too. But I don't know if you have a top 5 list or something.
Well, I do. We have, one is what's so-called unmet medical need, which means there's really nothing that's currently there. I mean, right now we have many options to prevent cervical cancer. And the best really is screening and then vaccination. And so we're not working on that. I mean, there's still many people in the world who don't get those. And so we unfortunately still have metastatic cervical cancer. But the unmet medical needs, where it hasn't changed since when I went to medical school, are diseases like pancreatic cancer and high-grade gliomas, glioblastomas, where the survival hasn't changed, except for the very few who are lucky enough to be, diagnosed early. So in the case both of pancreatic cancer and glioblastoma, the incidence is going up, and then the actual relative mortality rate from those diseases is out of proportion because, the incidence rate basically equals the death rate for these unfortunate tumors. So for me, pancreatic cancer is the one I have prioritized because it's accessible more than someone who has an intracranial lesion, which is harder to biopsy. We need desperately research in both of these diseases. But I think we're going, as a translational proving ground for advanced, sophisticated engineered cells, that pancreatic cancer is going to be the sweet spot for us to focus on.
Well, then when I think about PEN, I also think about some groups there that have quite good pancreatic cancer models in the mouse systems, at least, that pretty well recapitulate human disease. So you could be well set up there too, from a practical perspective.
That's exactly right. There's the Von der Heide and Beatty labs that have detailed studies into the tumor microenvironment of those tumors, and the genetically engineered mouse models now quite closely mimic what is being seen both in pancreatic cancer and have predicted the emerging success of CD40 agonistic antibodies for pancreatic cancer. So, we've had the huge breakthroughs in checkpoint therapies, and now I think for the first time we're seeing advances in agonistic antibody therapies, and that's based off mouse modeling.
I think one of the main interests when one thinks of cellular therapies is CAR T cells versus in comparison to transgenic TCR cells, and the advantages that either of these molecules can bring. So you have mostly focused on CAR T cells, which many people might suggest are intrinsically not the best for targeting solid tumors. What would you say to those ideas?
Yeah, so that's a complex question, Brenda. In the end, I'll just cut to the chase and say I think we'll be using combinations of TCRs, probably targeting both class 1 and class 2 peptide antigens, and as well as CAR cells. And the reason for that is much data now, both in humans and mice, shows that tumor cells learn to escape detection by cytotoxic T cells, either through loss of class 1 molecules or processing machinery. And if that's the case, then a CTL, unless you can restore class 1 presentation, won't work. And a CAR molecule then targeting another molecule independent of that then provides an orthogonal way to have where that escape mechanism would no longer work. But the other reason I hold out hope for CAR T cells in solid tumors is if you take tumor cells from patients, so and we've done this. It's not been published, but many different cancers from our tumor banks here at the University of Pennsylvania. We have, if you take the tumor cells and in a petri dish have CAR T cells that you add in, and you look mano a mano, what happens? The CAR T cells win. In other words, they kill these chemotherapy refractory tumor cells. And, this is then when there's no tumor microenvironment there. And there's no issue of trafficking. So if you can overcome those barriers, at least, a CAR T cell is very effective at the similar effector to target ratio at killing a pancreatic cancer cell as it is in killing a leukemia cell.
Interesting. And so, Philip, I know you've been, some of the struggle with your work is getting the constructs in, adapting the technology forward. I was wondering if you could speak to that, and also kind of the challenges down the road. Having switched industry myself recently, I've kind of learned about the difficulties of GMP manufacturing and all of those joys. I know Penn is good at, especially for an academic institution, very good at it, and part of what the CAR T therapy program built out. And so I'm wondering if you're thinking about those challenges and how, targeted payload therapies work in that kind of somewhat burdensome environment.
Yeah, thanks. It's a very good question. So in my case, since my idea was to kind of design more complex genetic circuits for CAR T cells, the underlying issue there is that these things become very, very big. So currently, what we put in a CAR T cell is, or what we put in a human T cell is essentially just a receptor, if you will. So the CAR receptor. But if you want to make it more complex, you have to put genetic switches like additional receptors or promoters that are turned off and on under certain circumstances. So you have to design like an entire circuit, and that becomes very, very big. And the lentivirus that we currently use to generate CAR T cells, they're all based on wild type HIV sequences. And the wild type HIV genome has a certain size. I think it's around 9 point something kb. So if you try to pack something that is larger than the wild type HIV genome size, you get a huge drop in efficiency. So your lentivirus titers drops a lot, and you will be able to make CAR T cells that or you will essentially, once you infect your T cells with virus that you made with this large construct, you will get only 1 or 2% of CAR positive cells. And this is not translatable to the clinic, because if you make a product where 99% of the cells are not CAR T cells, then that's not useful. So one of my first things that I did, I actually spent almost a year doing this, which is not the most exciting work, because it's not giving you any hard data, but I had to optimize all of the protocols from cloning to lentivirus design, lentivirus production, CAR T production. I'm actually hoping to publish a small methods paper in the next half a year. And I was able to kind of, increase the yield from I think the first time I put one of my large designs in a CAR T cell, I got 3 or 4%. It was very sad. The cells actually worked, but it was so few cells, you could barely do any experiments. Towards the end now, I'm getting 25 to 30%, which is very, very good. So I think it's, of course, more would always be better, but it's enough to do, in vivo studies. And that's one of the big things. I think that a lot of labs are now pursuing the idea of co-expressing additional receptors, maybe switch receptors or synapse receptors, or co-expressing cytokines. But all this will come down to the same issue, that as long as you're using lentiviruses, you run into the size limitation. So you have to optimize your protocols, or eventually we will switch from lentiviruses to other systems also. But I think once you stay with lentiviruses, you will have that issue with the size, and that's why you have to optimize your protocols to really be able to do experiments and translate this into the clinic.
That's really awesome in a way, though, because you're able to stay with the same lentiviral construct, it sounds like, right? The base one that is the GMP certified system, so that you're not having to redo that whole supply chain down the road.
And that's also one thing that kind of I learned, in in Carl's lab is that, Carl is very much focused on translation. So when you speak to him, he always talks about, okay, what's the benefits, eventually for the patients. He's always focused on, how can we get this into the clinic and into the patient, which was new to me because I come from, basic immunology where it's more, okay, how can we do the experiment or how can we get the paper out maybe? So there was, so that changes your mindset a bit because you can do like a lot of fancy things. You can, infect this T cell with 3 different types of vectors and make a lot of cool constructs, but in the end, it's not going to go into the clinic. So you always have to, you can come up with all these things, but you have to think about what can really get into the clinic, what can be, manufactured in a GMP way, in a way where you can use low MOIs, to infect the T cells. If you use too much lentiviruses, I think the FDA is not going to approve of that. So it's, you have to kind of stay within certain, restrictions so that you can actually translate it. And yeah, that's also something that is really a focus of the June Lab, that everything is, that we do should go eventually into humans. And that's everything that we kind of design is designed in such a way that it's more streamlined.
Yeah, I just would like to jump in on to enlarge something Philip just said. So in the pharmaceutical industry, but not the academic world, is the concept of a target product profile, or TPP. And this is looking at the end product. What would it actually be used for? And it helps if you guide your research towards something, in our case of a leukemia drug, it was something that would be able to kill, chemotherapy refractory cancer, but be able to be used in patients who've already had a lot of chemotherapy and have other issues. And so basic science research does not begin with a question of what the target product profile is. It's unrestricted research. But I find it very useful to try to couple the basic research with the target product profile. And then it's more likely to be translated and then actually be used in patients.
Talking about translation and the importance that these therapies have for the treatment of these leukemias that before were really hard to treat, and many of them would relapse, and patients had extremely low prospects. What is how do you see, for example, the future of CAR T cells when it comes to accessibility and the fact that nowadays it's really hard for, these therapies are licensed therapies are very expensive, and many academic centers are providing them and at lower prices, but at commercial prices is very hard to access, and a lot of people don't have access. How do you see a future in which more people can benefit from these therapies while still supporting the innovation and the funding from the pharmaceutical companies that in the end are payrolling a lot of this?
So, Brenda, it's a really complex issue. In general, if you look at total cost of medical care, and there, as you know, some large issues between single payer systems that you have and often in Europe versus the third party system we have in the U.S. But up until recently, the total cost actually for a cancer patient in care over the lifetime of their disease, only about 20% of that was from the cost of the drug. The rest of it was hospitalizations and all the issues that come up with patient visits and so on. So currently now with CAR T cells, the cost is much higher, and that's currently given at the end of many different therapies. So they've done economic analysis and shown that if CAR T cells are given, even at their current cost of around $500,000, depending on the setting, is more cost effective than what we currently do, if you look at the number of years the patient lives. So we now give a lot of therapies that fail or maybe give 6 weeks of additional life. Those are very expensive. And so that's the current economic value proposition. And if you move the CAR T cells up front, then you could not have all the other costs of what are failing therapies and actually cause side effects. So there's one thing, which is a political issue of when to integrate the therapies. There's a trial in the United States opened and running now by the Children's Oncology Group, where instead of right now, the FDA license in the US, and I think it's similar in Europe, is that CAR T cells are given for basically refractory and relapsed cancer twice. So you have to have gone through two different kinds of therapies, which are very expensive and toxic. Then you can give CAR cells. So there now is a trial testing, can you give it first line? And this is reserved for people with, by the genetics of their tumor, have high risk. And it's going to compare then the outcome of frontline cell therapy to standard of care, which is expensive and as well. So that then would lower the economic burden. But then I have a great faith in engineering sciences that, the cost of CAR T cells will come down by an order of magnitude through improved cell culture methodology. And then the big issue is, could so-called off-the-shelf CAR T cells be manufactured, from healthy donors, or iPSC is another source, or cord blood. And then that would take it down by another order of magnitude. So these things unfortunately do take time. The clinical trials take longer and longer when you treat earlier stage patients. So right now we're at this kind of unfortunate time early in a technology where it's more expensive. And it was the same way for cell phones. They were initially very expensive and came down in price.
I guess this is a also general topic or theme in immunotherapy in general, because I think the more and more, also when it comes to checkpoint inhibition, more clinical trials are looking at prioritizing immunotherapy over chemotherapy, and not only treated refractory patients, because also it seems like as chemotherapy also can affect the immune system of the patient negatively, it makes sense to, if possible, move up these therapies. They might actually even be more efficient, more effective against tumors. So I think as immunologists, it's very exciting to see, people not being just poisoned with chemotherapy, but maybe given other real choices that might harness their own immune system.
Yeah, there is this whole emerging new concept of cancer interception, which is the idea of identifying people at risk for cancer, and then through targeted therapies such as vaccination and hopefully cell therapies, preventing cancer from ever appearing. So that could happen in, for instance, in patients who have HPV infection. If you could install an immune system, it could prevent that from ever emerging into a cancer. You could also enable this in the setting of people with DNA repair defects, such as BRCA1 or BRCA2, familial histories of breast or ovarian cancer, and then instead of having surgery in those patients to prevent the cancer, it could be a vaccine or a cell therapy. And even then, there are, this huge thing now of things like circulating tumor cell detection and so on, where people could be, people at risk, say they smoke or whatever, they have environmental exposures, identifying those people and then treating them before the cancer is ever detected. So, those kinds of approaches, I think, will revolutionize how we currently treat, which is unfortunately, late stage cancers.
Yeah, no, I think it's interesting to see where the field's going. I think, being from the GI realm, seeing, colonoscopy is one of the early examples of surveillance. And even then they're moving from that type of approach to being able to see circulating cells. And then what can you do next, especially for the familial diseases. But to kind of shift over and talk a little bit more about careers and science life and such, you guys have both had interesting stories. I think the first question I had is for Carl, though, is that you spent a lot of time in your training in the military. And I trained, academically, clinically in the non-military setting. And so I was wondering if you could speak to how military medical training and thus your research training and all of that kind of formed you as the scientist and clinician that you are today, and how that's helped enable the work that you did to get done.
Thanks, Jason. That's it turned out, I came into the military because of the Vietnam War, and ended up having an obligation, for 12 years after they paid for all my medical school and undergraduate education. And the military has, unbeknownst to a lot of people, one of the best translational and infrastructure in existence outside of the pharmaceutical industry. So I came in, to the military and was not allowed I was trained as a bone marrow transplant physician for leukemia, but I had to do research in HIV because, and I also did research on malaria. The major research portfolio in the military is either for combat casualty care, trauma, TBI, et cetera, and also infections. More people in Vietnam lost time in Vietnam due to malaria than they did to enemy casualties. So, and that's true throughout war in general, is that infections are a major problem, dysentery and so on. So I worked on malaria and then HIV. The first published trial of a vaccine that had any efficacy in HIV was conducted by the military. And I was part of that infrastructure. I had no role in the science, but they did a trial of 17,000 people being vaccinated in Thailand when I was in the military. And, that was a huge infrastructure to do that. Now we benefit, the military is part of the U.S. vaccine consortium for, how the Moderna and Pfizer vaccines were tested so rapidly. So I learned the translational infrastructure while I was in the military. And then when I went to the University of Pennsylvania, I was able to apply that so that we could test engineered cell therapies.
And I think I could actually add to that because, when I speak to, friends and colleagues from other places, they all, they always say that the CCI is kind of a translational factory. So it's a clinical trial factory. And that was what Carl has established. Is very unique because you have tons of people that basically work behind the scenes, or that don't work in a lab. They have jobs outside. Basically, they are all there to kind of get the product once you know that it's working, to get the IND and make it go into a clinical trial and get it approved. So there's a whole setup. So the idea is always that when something is working, that's at least the impression that I have, that when you have something working, you have a very short way towards the clinic. Like I like in the labs that I've been so far, I think my last lab is an exception. So I worked previously in the lab of Michel Nussenzweig, and he actually, similar to Carl, he did something that people thought is cannot be done. He used to treat. He showed that you can use protein neutralizing antibodies to essentially suppress HIV viremia. And he also, I was fortunate that I was in his lab when his research translated into the clinic and they had clinical trials. But normally that's something very rare. Like when I speak to friends or colleagues, they never get to witness actually their scientific babies getting into a patient. And in Carl's lab, it's been exceptional. We have two students in the lab that made their own companies. We have several people that worked on projects that are now in clinical trials or actually in humans. So I think that's it's a very special place to be, the center, CCI.
What are the current clinical trials running at the CCI that you would like to highlight that people should be in the lookout for results?
Yeah. Unfortunately, the results take longer. I mean, we have many trials underway now, and some in the patients who don't respond to commercial CAR therapies for B-cell tumors. And, those look very promising. One of the issues has been with for instance, chronic lymphocytic leukemia, they're very much immunosuppressed, and maybe only half of the patients can be treated with CAR cells, and so that only about 20% basically get cured. So there's 80% that we can't really have an effective therapy for. We just have opened a trial with CAR T cells that also secrete interleukin 18, which then recruits the innate immune system through natural killer cells and macrophages. And so we're very excited about that. We have our first off-the-shelf CAR cells using CRISPR engineering, from healthy donors, and that leverages a recent trial we showed, the first time that you could use multiple guide RNAs and edit cells. And then we have ongoing trials in pancreatic cancer that I mentioned with many different strategies that should be open. We were unfortunately slowed down by COVID. The translational research slowed down much more than our bench science. So that unfortunately, I think, has hurt the whole field.
And I think all of clinical research everywhere, pretty much outside of COVID, was slowed down by COVID in the last year, unfortunately.
But because for our listeners, so far, all of the approved therapies with CAR T cells are aiming at CD19 expressing leukemia, lymphomas. That's the only approved therapy so far, right?
And, the field will be, refractory leukemia in the U.S. is about 5,000 patients a year. And I think all the blood cancers will have engineered cell therapies, both TCR-T and CAR-T, in the next, within the decade. The biggest unmet medical need has been myeloma and then acute myeloid leukemia. And myeloma is well known, is likely to have FDA approval of BCMA targeted CAR cells by, several of the big pharma within the next year. And that will be huge because myeloma is about 30,000 new cases a year in the U.S. and a very awful disease and very expensive to treat with non-curative therapies at this point. So we're excited to see, there's new indications for different kinds of CD19 CARs and there's many different B-cell malignancies.
Let's hope these new therapies can find their patients and can be approved and increase the availability of treatments for these patients. I have to say also, Carly, it's very comforting to see how much a person in your situation really cares about patients. And I have recently seen a presentation of yours, and it's how much you remember all these patients that were also the pioneers, the first one who got treated, and how much you also appreciate the risk that these patients are taking, especially at the beginning with this what start as experimental therapies, but then end up changing the landscape of treatment for these therapies. It's really great to see a lot of work being done to help these patients.
Yeah, thank you, Brenda.
Yeah, I can actually add to that. So that's one of the things that actually drew me, into the lab, because when I was actually at the point of applying to Carl's lab, I've been in the US already since 11 years by now, and at that point I was just finishing my PhD, so that was 3 years ago. So that was like 7-8 years, and we actually we were on the brink of do we go back to Europe? Do we stay in the US? And I had an epiphany moment during my PhD where I think it was 2015, where actually Carl came to Rockefeller and he gave a talk where he showed the results of the small clinical trials that they had back then with a few patients. I don't know, it was 15-20 patients, and the response rate, they were like phenomenal. And he gave this talk, and I was just blown away. I remember that this was one of the few talks that got me really excited, and I basically said, okay, I have to finish up what I'm doing currently, and this all makes sense. That's what I want to do. And then this really changed my trajectory kind of, and I said, okay, once I'm finished, I'm gonna apply to his lab. And I only applied actually to Carl's lab and one other lab, and Carl got back to me very, very quickly. We had a phone interview, and then I joined the lab basically as soon as I could. And one of the things that struck me also from his presentation was that he was so really like you said, connected to his research, because I think in science, people do science for different reasons. There are like scientists that like the competition, or they like just the discovery of something new. And then there are scientists that just really want to do something good, so that everyone, contributes. And Carl was so focused on, the patients and, doing something that really matters. That was just, exceptional for me.
It's great work, really admirable. Maybe as we are at the end of our interview, and continue with you, Philipp. So I wanted to ask you a question a little bit to get to know you a little bit better. And what is the best piece of advice you've ever been given, which can be professional or in non-professional realm, that you can think of?
So I would say it's an advice that I kind of didn't receive directly. I kind of came up with it together with a friend. We were both in our PhD, and we were working hard, and we basically said that doing science is like playing the lottery, and all that hard work does is you can basically buy more tickets, but it doesn't mean that you're actually going to win. Because if you look throughout science, you see people that win the jackpot with just one ticket, maybe a little effort, and you see people that work hard, buy tons of tickets, and they never get lucky. So that in the end you always need a bit of luck in science. That's just the reality of science a bit.
So Carl, my question for you is, if you were not a scientist or a physician, what would you be?
Well, that's a good question. I became a scientist and physician actually by accident. I thought I was going to be a chemical engineer, which is what my father was, and then the Vietnam War happened, and so I ended up in the military, and then actually got medical training. And so I was really fascinated with chemistry, and when I was in college, but before that, I wanted to be a professional athlete until I found out I wasn't good enough. But so I have a fascination with bicycle racing, and I'm still very active in that. But I think one advice to the people in training is, you don't really know what you're going to do. It will change, because you get different opportunities. And we have now this amazing time. It's a real gift from COVID, actually, that the society is realizing the value of science. And we need to continue that they understand how science can impact on people's lives. So I think there's a really bright future ahead now for science research.
So it's a silver lining in a cloudy, what's been a cloudy year. But I just want to take a moment to thank you guys for coming on with us. It's been an absolute pleasure.
Thank you for having us.
Yeah.
It was great. Thank you.
That brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and a link to all of the interview and Roundup papers. You can also reach out to us on Twitter at @immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests.